Hi guys, Miss Francis here. We left off discovering that DNA was the substance that carried our hereditary material, our genetic instructions. So today we're going to take a detailed look at how that DNA, that genetic information, replicates before a cell divides. So how long does it take DNA replication to occur? Well, it depends if it's a prokaryote or a eukaryote. In prokaryotes like E. coli, it takes less than an hour to do DNA replication. In humans, an example of a eukaryote, it takes a few hours. Regardless, this process is remarkably accurate where there's only one error per billion nucleotides. Now, where exactly does this replication occur? It happens at something called the origin of replication, but where in the cell does DNA replication occur? It occurs in the nucleus when we're talking about eukaryotes, and it actually happens during the S phase of interphase. So just to jog your memory a bit, cell division, mitosis, and DNA replication don't occur at the same time. Remember our cell cycle where we discussed the 24-hour clock? That interphase took up 23 of those hours. Mitosis was just one of those hours. Well, it's during the S phase of interphase that eukaryotic DNA is replicating. So just like I said, the DNA molecule begins replicating at the origin of replication, and what it actually forms is called a replication bubble, where replication can actually occur in both directions until the entire molecule is copied. So in eukaryotes, there may be hundreds or thousands of origin sites per chromosome, but in prokaryotes, there's only one site of origin, and that's because prokaryotes' DNA is in one giant loop. So prokaryotes will just have one single origin of replication, but DNA replication will still occur in both directions. So what I want to do is annotate this image, show you a couple of animations just so that you get the 3D feel of the two-dimensional pictures that we're going to be taking in our note packet. We'll take a look at the key players and then we'll have a discussion so that you can fill out your note packet. So annotating this image, you see that I've indicated where you have your original strand that is broken in half and your new DNA strands that are based on the template created by that original DNA strand. You'll notice that we have what's called a leading strand and a lagging strand, where the leading strand is in the 3' to 5' direction, and this lagging strand is in the 5' to 3' direction. That's going to be important when we discuss the action of DNA polymerase. DNA polymerase is the enzyme that's going to add nucleotides to our original DNA strand. And now this DNA polymerase works or elongates our DNA only in the 5' to 3' direction. In other words, our DNA polymerase reads in the 3' to 5' direction, but it elongates or works in the 5' to 3' direction, where it can only add a nucleotide to that 3' end. Now, in this example, it's important to recognize that our replication fork is on the right-hand side. That means we are unzipping to the right. Also means that the direction of replication is to the right. That's in this specific example. That won't be the case in every example. So make sure when you're evaluating a question, you check out which way the direction of replication is occurring. So here are our main players: helicase, DNA polymerase, primase, and ligase. And you should recognize that all of these key players end in ASE. That should tell you something. It should tell you that these are all different types of enzymes. I highly encourage you to revisit these animations that show you how DNA replication actually occurs. So let's take some notes. Let's meet our key players. Well, first is helicase, and helicase is what actually does the unwinding. It breaks our DNA along the hydrogen bonds between the nitrogenous bases. So why does DNA need to untwist? Because we need to read that DNA. If it didn't untwist, we wouldn't be able to have access to that DNA template. DNA polymerase, more specifically DNA polymerase 3, is known as the master builder, so to speak. DNA polymerase 3 is going to add nucleotides along the DNA template strands, and it adds those nucleotides according to Chargaff's base pair ruling. Now, once again, DNA polymerase 3 elongates in the 5' to 3 direction. It reads in the 3' to 5' direction. Now, how does DNA polymerase 3 know where to start? It knows where to start because this guy, primase, creates something called a primer. So primase is an enzyme that creates primers. Primers are made up of RNA, and it's this primer that is recognized by the DNA polymerase as the starting site for DNA replication. Ligase is known as the glue. It's going to glue Okazaki fragments. We didn't get to Okazaki fragments yet, but just bear with me, we'll get to it. It's going to glue the Okazaki fragments. So it basically forms the backbone of the new strand of the DNA. In other words, it creates those phosphodiester bonds between the sugar and phosphate on the backbone of our DNA. So once again, I said that DNA in eukaryotes can have multiple origins. In other words, you can have multiple bubbles, so to speak, where you've got many replication forks all going on at the same time. This plays a role in the leading strand versus the lagging strand because our DNA strands complement each other, meaning that they're not identical and they are called anti-parallel. So even when our DNA is unwound and our strands are parallel to each other, one strand is in the 3' to 5' direction and the other strand is in the 5' to 3' direction. This is important, again, because of the way DNA polymerase works. Remember, it only reads in the 3' to 5' direction. So one strand is going to be called our leading strand and the other strand is going to be called our lagging strand. So how does DNA polymerase know which side is the 3' side and which side is the 5' side? The 3' side has this hydroxyl group. That's how DNA polymerase 3 can identify it. So let's go through some basic steps. First of all, you've got helicase. Helicase is going to unwind the DNA, and then these single-stranded binding proteins, sometimes they're even called SSBP, or sometimes they're called just SSBs. They are what enable the DNA strand to stay separated. In other words, it prevents the hydrogen bonds from reforming between our nitrogenous bases found on opposite sides of the DNA strand. Next, our primase creates a primer so that our DNA polymerase can recognize that primer and begin to build the new strands. Again, I've reiterated this 3 times now. DNA polymerase will only work or elongate in the 5' to 3 direction. That makes it difficult when you unwind the DNA and you end up with a lagging strand because this lagging strand is in the 5' to 3 direction. DNA polymerase can only read in the 3' to 5' direction. So now what you've got is you've got these little fragments that are going to form. These fragments are known as Okazaki fragments. Do you also notice that the DNA is being built in the opposite direction of the replication fork. It's being built in the opposite direction that our DNA is being unwound. That's an essential difference between the leading strand and the lagging strand. And then you've got our good old pal ligase that's going to glue these Okazaki fragments together by forming phosphodiester bonds between the sugars and the phosphates. So here's the big difference between the leading strand and the lagging strand. Well, first of all, let's evaluate this picture, because this picture is different from the ones that we've just been going through. Do you notice that our leading strand indicates that our replication occurs in the direction of the replication fork? In other words, in this picture, the overall direction of replication is to the left. In all of our other pictures, it's been to the right. So that's what I mean by you have to look carefully at the picture, because not each picture shows the DNA unwinding in the same direction. So the leading strand, which happens to be this top strand, has DNA nucleotides being added in a continuous manner to the template strand. In other words, that DNA replication moves in the same direction as the unzipping takes place. It moves in the same direction as the replication fork. Whereas on the lagging strand, the DNA nucleotides are added to the template in fragments, known as Okazaki fragments. And nucleotides are added in the opposite direction of unzipping. In other words, the opposite direction of the replication fork. So here's what I want to go over with you guys. Here is our origin of replication. Do you see how I can kind of separate this in half? And notice that I've got 2 different replication forks, where this top strand is a parent strand and this bottom strand is also a parent strand. They act as the templates. But notice, one of those strands runs in the 3 to 5 direction, whereas the other strand runs in the 5 to 3 direction. So do you notice that within each replication fork, I'm going to have a leading strand and lagging strands. And on this side, I have my leading strand and I have my lagging strands. That's something to be mindful of. Take a look at this question, see if you can answer it. If you want, pause the video and see if you get it correct. If you answered letter D, good job, you got the right answer. So now, primer is added. Don't need it anymore because I'm done with that section of replication. So DNA polymerase 3 is what added the DNA nucleotide to the original strand. Well, then in comes RNase, sometimes called RNase H. That's going to remove the primer, and then you've got DNA polymerase 1 that's going to replace that RNA with DNA. So how many primers are going to be needed for your lagging strand? 1 per Okazaki fragment. Whereas in our leading strand, I only need 1 primer because it occurs in a continuous pattern. It's uninterrupted. So again, I encourage you to revisit these animations to help you wrap your brain around this complicated process of DNA replication.